polymerase II is a transcription machine, but even machines stall. And one of the biggest obstacles in its way? Nucleosomes. They're like speed bumps made of DNA and histone proteins. So how does RNA Pol II keep going? A new mechanism reveals that two factors, NDF and FACT, team up and form tiny droplets that help the polymerase power through chromatin. Chromatin makes eukaryotic genomes compact and organized, but it also makes transcription harder. Every nucleosome wraps around 147 base pairs of DNA, and RNA Pol II constantly runs into these structures as it moves along a gene. FACT facilitates chromatin transcription, is a histone chaperone that helps RNA Pol II deal with nucleosomal barriers by partially disassembling the nucleosome. Specifically, FACT removes the H2A-H2B dimer, converting the nucleosome into a hexosome on the DNA. Once RNA Pol II passes, FACT redeposits the dimer, restoring the nucleosome and resetting chromatin structure. But here is the challenge. RNA Pol II moves fast, extremely fast, up to 3.5 kilobases per minute in human cells. So how does FACT reach the polymerase in time and function quickly enough to keep up? Meanwhile, research in transcription biology has shown that many transcriptional regulators form biomolecular condensates, liquid-like droplets formed through phase separation. They act like molecular co-working spaces, concentrating proteins exactly where they are needed. The study featured here reveals that NDF, nucleosome destabilizing factor, forms phase-separated condensates with FACT. These condensates assemble directly on elongating RNA Pol II and enhance its ability to transcribe through nucleosomes. So how do these proteins come together? Tandem affinity purification, followed by mass spectrometry in HeLa cells, identified FACT as a major interacting partner of NDF, and co-immunoprecipitation confirmed the interaction. As RNA Pol II encounters a nucleosome, it pulses at defined superhelical locations, most prominently at SHL-5-4 and SHL-1. NDF alleviates the early pulse, while FACT resolves the later one. But when both proteins are present simultaneously, nearly all pulses upstream of SHL-1 disappear, leading to a significant increase in full-length transcription product. Together, NDF and FACT function synergistically, enabling RNA Pol II to traverse nucleosome barriers with far greater efficiency. So how do NDF and FACT work together so effectively? An important clue emerged from an unexpected problem. Attempts to determine the NDF-FACT structure by cryo-EM repeatedly failed because the complex aggregated during purification. Unhelpful for structural resolution, but highly informative biologically. This behavior suggested that NDF and FACT, both rich in intrinsically disordered regions, might engage in multivalent interactions characteristic of phase separation. And indeed, purified NDF or FACT alone did not form condensates, but when mixed together, they immediately generated micrometer-scale spherical droplets at very low concentrations. The same phenomenon was observed with their yeast homologs, demonstrating evolutionary conservation of NDF-FACT condensate formation. Moreover, preformed NDF-FACT condensates rapidly recruited thionein-labeled mononucleosomes within just 10 seconds, distributing them evenly throughout the droplet. This rapid uptake is striking, because FACT alone interacts only weakly with intact nucleosomes in vitro, suggesting that phase separation creates a specialized microenvironment that enhances engagement with nucleosomes. To test whether NDF-FACT condensates assemble directly on a single RNA Pol II nucleosome complex, the researchers used a C-trap system combining optical tweezers with confocal fluorescence. With fluorescent labels on NDF, FACT, and the DNA tethered to RNA POL2, they saw both proteins aligned precisely at the RNA POL2 position. This shows that the condensate forms on RNA POL2 and rides with it as it transcribes. Finally, the study tested whether NDF FACT condensate formation matters in vivo. Using CRISPR-Cas9, researchers created human-induced pluripotent stem cells expressing the NDF K161A mutant, which still binds FACT and retains catalytic activity, but cannot form condensate with FACT. These mutant stem cells remained pluripotent, but grew noticeably slower than wild type. And RNA sequencing revealed a global reduction in steady-state transcript levels, indicating widespread transcription defects. To probe elongation dynamics, researchers treated cells with DRB, which is a transcription inhibitor that induces promoter proximal pulsing of RNA POL2, and then washed it out. Using transient transcriptome sequencing to monitor transcription resumption, they found that elongation rate was reduced in mutant cells. Together, these results demonstrate that NDF FACT condensate formation is essential in vivo, not just in vitro. Because FACT is linked to multiple cancers, insights into NDF FACT condensates may inform future therapeutic strategies. More broadly, this work shows how phase separation enhances transcriptional efficiency, reinforcing the growing view that gene regulation is organized within biomolecular condensates.